In today's lecture, we are going to talk about Sia and Dawson's demonstration of transformation in S. pneumonia. So, Sia and Dawson conducted a similar experiment as that of Griffith in 1931 in vitro setup. Griffith's experiment was an in vivo setup. Note the difference that in vivo means it was done in the mice, as you might remember from my previous lecture. They showed that transformation could be carried out in liquid cultures of S. pneumonia and that mice had no role in transformation. And it was a further validation step of Griffith's experiment. Why they might have conducted this experiment is that they would have had a doubt on how transformation is taking place or if the host had any role in it. So as we move further, I'll be explaining this entire setup and the conclusion so drawn. So they had basically three sets of experiments in which the first setup consisted of living R-type strain which was grown in the culture media and further inoculated onto agar plate, and he found R colonies. It's just as similar, just assume, this no, you're not supposed to assume. It's this culture media is analogous. It's an in vitro analog of your mice. Okay. And similarly, the heat killed S strain when they grew it in the culture media and further inoculated on the agar plates, they find no colonies because the S strain consisted of this capsule, and as you give the heat shock to bacteria, the polysaccharide capsule is destroyed, and that's why you can't see any colonies. Here comes the third setup. In the third setup, they grew R-type live R-type with heat killed S-type and further precipitated the R-type from the broth just to distinguish which R-type strains might have taken the transforming principle from S strain or not, just to distinguish whether distinguish the transformed ones from non-transformed ones, and further when the group inoculated them on culture plates, they found S colonies. So now it was validated that although S type was heat killed, and its polysaccharide coat got destroyed, but its genetic material was intact, which was taken up by the living R type, which earlier Griffith called as the transformation, and mice had actually no role in this process. So that's all for this experiment. Hope you liked the video, and all your feedbacks are welcome in comment section. Thank you.